guys. It's been a while since I've made a video, so hopefully this works out well. I'm also trying a new thing by doing it in QuickTime, so that's new for me. So let's see how this goes. If you've got any questions, send me a remind text or feel free to ask when we're back in class next week. So quick, this is our Introduction to Genetics unit. This is replacing the lecture that I'm not doing in class today. So, here goes. First thing I want to do is I would like to review meiosis, which again, we just spent some days on that. But the gist of meiosis is you have diploid parents. So you yourself, all of your body cells are diploid. They have 2 times the number of chromosomes that are found in our gametes. So that when we produce gametes like sperm and egg, those are haploid. They have half the number. So that when 2 haploid gametes come together, when the sperm fertilizes the egg, you now have a diploid number of chromosomes in that particular offspring. So that's kind of an important thing because we're going to end up talking about gametes a lot today, just so you know. All right, so some review terms. First, we have alleles. Alleles are particular forms of a gene. So that's like which copy of a trait did you get? So you got 1 copy of a trait from mom, 1 copy of a trait from dad. And so depending on what trait you actually inherit, which allele you get, your allele might be a dominant allele or it might be a recessive allele. So if it's a dominant allele, that means that trait is going to show up. Whether you have 1 copy of it or 2 copies of it, you will see that dominant trait. If you inherit the recessive trait, you only will see the recessive trait if 2 of them are present. So I kind of think about it like the buddy system. You will only see those recessive alleles if they're traveling in the buddy system and you inherit 2 copies of the recessive alleles. Some other terms, and again, these terms I'm thinking are review terms. If they're not, you want to make it so they seem like they're review terms. So study them, flashcards, read them a bunch of times, whatever. The next 2 words I like to pair up are homozygous and heterozygous. Again, the word part homo means the same, hetero means different. And so what we're looking at with homozygous is you have 2 identical alleles. It doesn't specify whether it's, capital R, or the 2 dominant traits, or little r, the 2 recessive traits. But homozygous just means that those 2 letters are both the same. Those 2 alleles are both the same. If you have heterozygous alleles, that means you have 1 dominant copy and 1 recessive copy. So 1 big R and 1 little r. Those are different. When we're talking about homozygous, you can be homozygous dominant or you can be homozygous recessive. You do need to specify what type of homozygous you are, but you can just say heterozygous if you have 1 copy of each. Because if you are heterozygous, you appear dominant because you have that 1 dominant trait and it really shows up because it's dominant. All right, couple more words. And again, looking at genotype and phenotype in pairs. A genotype is the particular combination of alleles that you inherit. What I'm trying to teach this for the first time, and I want people to really remember, I say genotype is what the genes say. So it's actually what those alleles code for, the letters is kind of a way to think about it. Versus your phenotype is your observable effect of an allele combination. So that, again, putting it into me, your phenotype is your physical appearance. So phenotype, physical, geno, gene say. Anyway, that's how my brain does it. So when you're talking about phenotypes, you're talking about physical descriptions, whether someone has blue eyes or the plant has pink flowers or wrinkled seeds. So those are your physical descriptions versus for pink flowers, you may have a big P and a little p, or you may have a big P and a big P. Those might be your genotypes, but the phenotype is pink flowers. The last word we're going to talk about kind of from now on through the rest of the year is frequency. And that's kind of another way to say percentage. So when we're talking about frequencies, you could talk about the frequency of alleles in a population. You could talk about the frequency of a genotype in a population or the frequency of a phenotype in a population. So, question, like a trivia question, I guess. In a population of roses, 0.3 are pink, 0.15 are white, and 0.55 are red. So are we talking about the allele frequency? Are we talking about the you know, particular traits that exist in a population? Are we talking about the genotypes? Are we talking about what the genes say, what the letter combinations are? Or are we talking about the phenotype frequency, what the physical appearances are? Hopefully you picked phenotypes, physical appearances. All right, I do want to remind you, because I forgot at the beginning, if at any time you need to pause, rewind, replay this video, please do that. I won't ever know. And that way you can stop and write down what you need to write down. All right, let's go. Genetics, woohoo. Okay, so a long time ago, back in the 1800s, the scientists just sort of thought everything blended together. And so if you had like a red flower and a white flower, then obviously your babies were going to be pink. And an example that people see and they think is like skin color. Skin color is crazy complicated. But if you have someone that is super, super pale and someone who has darker skin tone, they may have a baby that's in between. And so that made sense for blending. Except this cool guy, Gregor Mendel, Mendel's my Chrome boy. Gregor Mendel says, no, guys, it's really not like that. There's dominant traits and there's recessive traits. And so he did a bunch of research on pea plants and snapdragons and all sorts of different things. Like he was a botanist. He was into plants. So anyway, he says principle of dominance. He says certain traits are dominant, certain traits are recessive. And unless you have the two copies of the recessive trait, you only see the dominant trait. Red is dominant to white. I have a red flower and a white flower, and they love each other very much. And they decide to have baby flowers, and those baby flowers turn out to be red. Really fast, this is called the P generation, the parents. And then the F1 generation would be that first offspring that's produced. It stands for filial, so it means the offspring. So the F1 generation are the first offspring produced. Okay, so the reason why this can happen and we can have these different combinations have to do with the fact that you inherit two alleles. One allele comes from mom, one allele comes from dad. So if you inherited the dominant F allele, let's say the capital F, from mom and the dominant F allele from dad, then you're homozygous dominant. You have two copies of that dominant trait. Your phenotype is the dominant trait, which in this example is a red flower. So then if you are heterozygous, you inherited one dominant copy from a parent and you inherited one recessive copy from a parent, big F, little f. Well, because you have even one of those dominant traits, you appear dominant. And then the third one, my third example on the far right, is when you inherit a recessive trait from mom and a recessive trait from dad. If you get a recessive trait from both parents, just like the buddy system, you are going to appear recessive. And in this example, that means a white flower. Homozygous recessive is what you would call those. So here's some just silly examples. Awesomeness is a dominant trait, shown with the letter capital A. What is the phenotype of someone whose genotype is little a, little a? Think about it. Think about it. Pause if you don't know the answer. But if you do and you picked not awesome, then you'd be right. So what would we call that genotype? What are the words we would use? Hetero or homo? Zygous, heterozygous, or homozygous? I'm going to assume you also said homozygous. Homozygous recessive specifically. All right, grumpiness is a recessive trait shown with the letter G. What is the phenotype of someone whose genotype is big G, little g? Well, hopefully happiness is the opposite to grumpiness. And since they have one dominant trait, they are going to appear happy. Okay? And so that genotype is considered to be heterozygous. Punnett squares. I feel like almost always this is like the favorite thing that everyone always remembers from, back in their middle school classes. And so I'm just going to hope that that's the case too. If not, we'll talk. But here's the quick overview of how these things work. So first of all, on the outside of your Punnett square, you're going to set them up like a little 4-square box. And you have the gametes from both parents. So parent 1 I've got sitting on top, big R, little r. And parent 2 I have over to the left side, big R, little r. And so then from there, we fill in the boxes. And when we fill in the boxes, that tells us the genotypes of all of the possible offsprings. So it's actually pretty easy. So this one pretty much drops on down here, and then we bring the big R in from the left, and then we get this box where we have big R, little r. Well, that was weird. I didn't expect it to do that. So we've got this big R, little r, and then the same thing would happen where big R would go to the right-hand box, and the little r drops on down. And so we're able to come up with what all of our genotypes would be for all of the offspring by just kind of filling in the table. And so the cool thing about Punnett squares is we use that to calculate probability, to predict what we think the offspring's, what percentage we would have of different types of offspring. So in this case, the purple coloring is showing our 50% chance of having a heterozygous offspring. The big R, the green one, is showing us a 25% chance of having a homozygous dominant offspring. And then the little r, our red one, is showing us a 25% chance of having a homozygous recessive offspring. So I want to make sure that you're okay using these words. I suspect when you see questions about this in the future, it will involve the words and being able to use words to describe genotypes and phenotypes. All right, so here's some really cute guinea pig examples. And so again, pause as we go and make sure that I would recommend you work this problem out with me. Anyway, so here we have guinea pigs, short hair. So the cute little guinea pig on the right, which by the way came from a guinea pig daycare website, is homozygous, is dominant to long hair, little h, cute guy on the left. So across a heterozygous short-haired guinea pig with a long-haired guinea pig. So we have to decide first what gametes will each one produce, and then what percentage of the offspring will be big H, what percentage will be big H, and what percentage will be little h. And then once we figure that out, we have to come up with the phenotypic frequencies. So what percentage of the offspring will have short hair and what percentage will have long hair. So how do we do that? First, let's tackle the first part of the problem. All right, if you are heterozygous, that means the traits, the alleles you could pass on a big H to your babies, or you could pass on a little h to your babies. Those are your only choices. And since you aren't giving your babies both of those, you're only giving them one, you're producing gametes that only have one allele in them. The long-haired guinea pig has the ability to only pass on the little h. And so all of the gametes that that guinea pig makes will be little h. That will be the allele that they can pass on. So then when we set up our Punnett square and then we fill in the boxes, we see that none of our babies, 0% of our babies have the genotype big H. But half of our babies, 50%, 2 out of 4 boxes, have the genotype big H, and 2 out of 4 boxes, or 50% of our offspring, have the genotype little h. So what are those phenotypes going to be? Well, if they have some that are dominant, that have, that dominant trait, those are going to be short-haired. So 50% of our offspring have that dominant trait, even if it's only one copy. And then 50% of our offspring do not have any dominant traits. They only have the recessive traits, so they're going to have long hair. I highly recommend you take a look at these practice problems, especially if you are not 100% confident with how to do Punnett squares. This is super easy practice. If everything goes well, I'll try and post these answers for you. I am going to pause this video here. I'll consider this the end of part 1. I want to talk about dihybrid crosses next, but I'm afraid I'm going to run out of time on YouTube. So pausing here, and then the next one we'll be talking about dihybrid crosses. All right, thank you. Bye.